Okay, we're live now. Should be. Yeah.
Sorry. Yeah, it's working.
Awesome.
Yeah, just got a notification on my phone that OoLEN is live.
Okay, then, Siddhant, the floor is yours.
Should we wait a bit for people to join in? I believe we can give it 2 minutes for people who are fiddling with the link and finding it.
Yeah, I'd say that's a good idea, yeah.
So yeah, people are definitely joining in. Hi, Silvana. Okay, I think we'll start. Hello, everyone. Welcome to the first public OoLEN meeting. And today we'll have 2 people with us who are going to discuss their science as well as their career transitions, whether it's a PI, whether it's a postdoc as an early career researcher in origins of life. So first, we have Martina Preiner and we have Blokhuis. Martina will go first, and she'll be talking about finding transitions from geo to biochemistry. And yeah, she'll be introducing her, but as a good host, I should give some few lines. So Martina is currently a Max Planck research group leader at the Microcosmo and Earth Center at MPI, Institute for Terrestrial Microbiology in Marburg, Germany. And she's a very new PI. She started her group in January 2023. And the floor is yours, Martina.
Yeah, thank you so much. Also, thank you so much for inviting me here. It's really exciting to be in this very first meeting seminar of this kind. And as Siddhant said, I'm quite a new group leader. Actually, I mean, I started in January, but actually the group just started a bit in March. So we're still in the very first steps. But as I was asked to talk a bit about my career, or what you call a career, whatever, I have a very boring way to do so. I just wanted to show you. I just have to click in here, I guess. Yes. So what my actual CV is like. So it has been a while since I started and finished my bachelor's. I started in 2004. It was in biochemistry and chemistry in Munich, so in Germany. And I did a bachelor thesis on fluorescent proteins, so basically more biochemistry than chemistry. Then I did a master's in chemistry and focused on physical chemistry, looking at protein folding, so it was biophysics in that direction. But while I was doing my bachelor and my master's, I liked chemistry, but also, as you might see, what I chose to do in my bachelor thesis, I always kind of felt like, I'm actually also kind of interested in neurobiology. How am I stuck, so to speak, in chemistry for all this time? And the second thing was, I felt, especially during my diploma, already a bit restless. I felt like it was too much focus on one thing. And I started in a science department of a local student radio station in Munich. That was very lucky. I started that during my master thesis. It was nice of my supervisor back then to actually give me 1 day a week where I could go there. Try to make up like on the weekends, etc. But in the end, it was nice enough to get this opportunity. And that's also how I realized that I was interested in science in a more general way, not only in like doing science in academia. So I kind of stumbled a bit into a mentoring program of a German initiative for science journalism. And there was roughly a year where we were trained. We were all mostly natural scientists, 12 of us in total, where we were trained in how to actually use the science and develop journalistic background. How do we learn to write? How do we actually learn how to speak on radio? How to write articles? How to write broadcasts? Also, in theory, at least, how to do TV, etc., which was really lucky for me. And from that time on, I used to be a freelance science journalist for quite a while, 5 to 6 years, for German media mostly. So as a natural German speaker, as you might hear from my accent. So I did a lot of different things. I also was not focused on one science. I actually reported a lot on neuroscience, because neuroscience was, I mean, not what I studied, but what I was mostly interested in. And to be honest, like rough chemistry usually, at least back then, was not what people were most super interested in. So I did very different pieces, different backgrounds, different sciences for that, and also different media. So I did radio, I did TV, online and print. So I tested everything and I loved everything equally. And then afterwards, I have to admit, I got a little bit tired of the freelancing and tried out if I could also do more permanent positions, also in PR a bit, or also in production companies, which means you also get fixed money, so to speak. Just to give you a background, what I felt during that time, when I started out, I felt like super amazed. It was great to meet those people who are feeling alike, you know, like I don't want to be focused, like I don't want to be restrained by academia. I just want to feel science, report on science, also be critical towards science, and not just like take it as something that you have to sell, but actually rather that something that you also think about a bit. So, and I felt when I became a journalist and I actually made money out of it, and great, like not a lot, I have to admit, but enough to live from it. I really felt like I found my dream job. So that's the thing that I was supposed to do. I could report on anything that was interesting. I didn't actually have to do the science. I could just like go to people that were successful in it or had something to say about it. And I also liked that I could put it in a perspective for society as well. So in the very end, though, not in the very end, but the longer I was there, also struggled with the freelancing, as I told you already. And I tried to find different paths. So it was also a downside after a few years. So it's not like that you find something and then you love it just for eternity. And what happened to me was actually, I also stumbled again onto something that caught my interest and my passion, because I interviewed a few origin of life researchers on a piece for a German online platform. And as a joke, kind of, I said to one of them, well, if you need someone to do this, because they were looking for a PhD student, I actually have a master's in chemistry. It has been a while, but maybe you still need someone. And that actually led to a PhD. And I did my PhD and also partly a postdoc at the Heinrich Heine University in Düsseldorf at Bill Martin's group, who was crazy enough to say, yeah, well, this chemist who hasn't been in the lab for 6 years, why not give her a chance? And started working on the origin of carbon metabolism there with a biologist. Also at the same time, I had an Audible science podcast with a friend, which was great. So I could still do some journalism as well. So it was kind of a really perfect match for me. But was also nice for me to experience was that I actually had to build up a chemistry lab or a chemistry niche in a biology lab. So I also had to find the fitting collaboration partners. I had and wanted to stay in different various other labs, which was great because I've met people, I met different disciplines, and I also met how friendly people can be if you actually ask them if you can learn something from them. What also was taking place during my PhD. I mean, we all know that, that's why OoLEN exists, is that the origin of life field can be quite restrictive depending on where you look. And Joanna, Javier, and I, we got a little bit sick of it. We were in the same group, and we wanted to organize something so especially early career researchers could meet and exchange their thoughts and their science on origin of life. And that's what we did in 2018, which then ultimately also, thanks to a lot of other people, led to the formation of OoLEN. But before that, that's also important to important part of me. I became a mom in 2020, so that also kind of led me into a different focus as well. What I did then was a postdoc in geosciences that was really interesting for me. I also got to do some field work from 2021 to 2022. I actually wanted to stay there longer, so my contract was 3 years, but then I applied to a position in Marburg in Germany that was open for a Max Planck group leader position. And first of all, I have to admit, I didn't even want to apply because I thought, well, it's microbiology. I'm not a microbiologist. Who's going to take me there? But luckily for me, I had friends and colleagues who said, no, you have to try, and your science could fit in quite well with that. And what happened, I just had moved to Utrecht, I think, like for 6 months. So I applied, not really thinking that I could have a chance to get it. I went there, and I actually saw that microbiology and my type of chemistry and prebiotic chemistry kind of fits together super nicely, and that I could really find a position there. And that also led kind of in a right place, right time situation. I got the appointment for this group leader position that I'm really thankful for. And as Silan also said, I just started out on that, and I'm kind of curious where it would lead us all. So I moved to Marburg in 2023, and why I'm actually having these parts, the moves, that basically has a little bit to do also with me becoming a mom, because it gets actually also more exhausting to move for any type of position. What I wanted to, my take home message, because I also want to talk about science, of course, and I already talked a bit too much about myself, is don't restrict yourselves to something that you think is necessary to stay in academia. I think that is my take-home message from also what my career was like. I did kind of what I wanted to do or what I was interested in, and it kind of worked out for me. And I don't have any regrets in that sense. Other, maybe the moves were kind of too much in the past 2 years. And it was also kind of exhausting for my partner, for example. That's also something to take into account. But this is, if you have any more questions also on not complete stringent career path in academia, please reach out to me. And also keep in mind that at any time and place of my career, I could have gone a different direction, and I think I would have been fine as well. That's maybe also something to take into account. There's more than just one path to go with what we're doing. So without further ado, again, if you have questions and there's not enough time later on, or there's maybe more private questions, whatever, feel free to reach out to me via mail or via text or whatever, and ask me any more detailed question on how everything went down. To answer the question that I didn't even comment before, I really was looking for myself on all my career process as well. So really figuring out who I am. And I think the most satisfying answer to that question that I have now is like, I'm a generalist. I like to do a lot of things. I might not be a specialist in any of those, but I really enjoy to be in the middle of everything. So that brings me now to what I'm doing now, what I used to do. And I have my group exploded kind of in the past 2 weeks. So I'm not sure if you can see my cursor, but maybe I can. Yes. So this is my group now, but for, I think, until mid-September, it was just Elfina who was brave enough to actually come with me to start out the lab and everything. But Oscar, Giuseppe, Amal, Mayop, and Zainab are also there right now doing their work. But what I will be focusing on, because I also cannot share science that is not really done yet, I want to focus on science that I did with the following people of the plethora of collaboration partners and friends I have in this field. So since I started my PhD in 2016, as most of us, I think here, I was fascinated with the question, how did we get here? So to single cellular organisms, I cannot say they're primitive because they're already quite complex at this point. From here, simple geochemical abiotic settings such as rock pores, earth crust, whatever you want to fill in from your hypothesis or the hypothesis you find more likely in here. So we know there's a top-down approach that's usually bioinformatics to kind of look for the most conserved enzymes, proteins, to make a theoretical construct of what the last universal ancestor could have looked like. I think it's just important to say at this point, it is to remind ourselves, it is a theoretical construct. It's depending on stepwise, we are getting closer how Luca could have really looked like. And from there, we hopefully can then make bottom-up approaches to find us somewhere in the middle. Also for the bottom-up approaches, of course, there's a lot of different things you could do. But what I'm focusing on, or also want to focus on, is kind of summarized to this. You don't have to agree with it, but it maybe helps you to follow the line of research that I'm trying to do. So if you have a porous environment such as the earth crust, water rock interactions, you would start with the geochemical processes that such as serpentinization, and we'll come back to that in a bit. From there on, you could make another step to catalysis on mineral surfaces, then quite a jump to self-sustaining catalytic networks, and from there I would see polymerization and first protoenzymes to develop. And this is a line that we're trying to follow with our research as well. So if you do those reconstructions from a top-down approach, you can come up with a possible physiology of Luca. This work was not done by me, but by colleagues in Düsseldorf, and they figured out that possibly or most likely Luca was an anaerobic autotroph, which translates into it was CO2 fixing and hydrogen and transition metal dependent, because that's what it needed for metabolism. And that means we can make deductions about the possible habitat of Luca, which meant it had to be geochemically active and also had hydrogen, CO2, and transition metals for the at least. Luckily for us, those systems that provide that still exist nowadays. They're called serpentinizing systems, and they come actually in many forms. They can be hot and acidic nowadays, or cool and alkaline. They're also presumed to be not only on Earth, but also, for example, here you can see Enceladus on icy moon around Saturn. And most importantly for our line of work, I think they're also considered ancient. So as soon as you would have water rock interactions, you would have type serpentinizing type of reactions as well. So just to zoom in a bit, what do I mean by earth crust? While I was doing my postdoc, we were also able to collect some samples from an actual hydrothermal vent field that is serpentinizing. And if you just zoom in into a rock sample like that, you can actually also find metals and metal sulfides, metal oxides that could be good catalysts for prebiotic chemistry. In those systems, we also find an alternation of low and high water activity. We have pores for compartmentalization and concentration of products. And we have hydrogen, very important as an electron and energy carrier, and also CO2 as a carbon source. So I'm not sure if I really have the time for that, but I wanted to take a little bit of more time to explain serpentinization. So it's a water rock interaction that most likely already happened 4 billion years ago, which is great. So that means the protons of water are being reduced in a process where a rock called olivine or minerals called olivine are being transformed into serpentinite. That is a process from iron 2 to iron 3, and the electron of this oxidation of iron minerals is being put on a proton of water. So you have the production of hydrogen, which is great because it is a great energy carrier, not only for microbes, but also, as we know, the latest since Hindenburg, that is a general carrier of energy that can also have quite violent reactions in the end, but nature is able to confine it and harvest the energy stepwise. So we have hydrogen now. What is happening if we're actually taking CO2 into account in these systems? That means we have the reduction of CO2 with the help of the electrons of hydrogen. We can also measure that in serpentizing systems. It's not only methane, it's also acetate, other organic compounds that we find in these systems that are actually being measured in the fluids. So the question is, if we have the CO2 fixation processes inside the Earth crust, is this reaction between CO2 and hydrogen maybe something that could have kickstarted protometabolism? Why is this reaction so important? From a microbial point of view, it's especially the Wood-Lungal pathway, or in other terms, the reductive acid-to-CoA pathway. Why this reaction is so interesting for several reasons, because the Wood-Lungal pathway is considered the most ancient CO2 fixation pathway on Earth. It's also the only exergonic one that we know of, and it's also the only linear CO2 fixation pathway that we know. So it's like a one-way street into metabolism, and it's, from my point of view, very interesting to look in more detail into the options of it. So nowadays, there are roughly, depending on what organism you're looking at, 9 enzymes and 10 cofactors involved in this process. And just that you understand it a bit, you have a methyl branch and a carbonyl branch in the acid-to-CoA pathway. So on the methyl branch, you have a stepwise process of reduction of CO2 to a methyl group, and on the carbonyl, like a one-step reduction to a carbonyl group, which will be unified to acetyl to an acetyl group here. And if you then go even further in the reduction, would get to pyruvate, and from there to oxaloacetate to the RTCA, etc. So you can build up carbon metabolism from that direction. So when I'm talking about the enzymes and the cofactors that are involved in the Wood-Lungal pathway, the ones that really fascinate me most are hydrogenases, for the simple reason that they actually make the electrons accessible to the metabolism, the electrons accessible to carbon. And this is one example. It's a nickel iron hydrogenase, and all the other hydrogenases actually have also transition metals and then active center. And when I started my PhD, that's also something we wanted to find out, if actually mineral surfaces could provide us with a protoenzyme, so to speak. So could mineral surfaces do the same as enzymes? So we tried out different minerals that you would also find in serpentizing systems, such as magnetite or nickel-3 iron called averroite. And what you can see here from our reactions, the products that we got, so formate, methanol, acetate, pyruvate, in different concentrations, but the same distribution, basically, of molecules, are the discrete steps that you also find in the Wood-Lungal pathway itself. So what we thought to see here is an abiotic pattern of CO2 fixation. There was doesn't look like much, but it was a huge collaborative effort between Japan and Strasbourg and Mülheim and Düsseldorf. And I'm really glad that that this turned out to be my PhD thesis in the end. So, so far so good. What are the next steps? I think 3-4 minutes maybe, but you can also tell me if I should shut up, Sidant. So the one big step that we have from like simple catalysis of mineral surfaces, or also homogeneous catalysis in solution, whatever, is to a self-sustaining catalytic network. How the hell are we supposed to get there? And I think a way, or not only I think that, but a lot of other people started thinking that as well a few years ago, and also previously before that as well, is that a possible way to connect those two things, the biochemistry on one hand side, or autocatalytic network on one side, and just geochemistry on the other, are cofactors. Here you see a few examples. So it's one of the most fascinating hydrogenases that I know of, the iron hydrogenase, because here you can see 2 organic cofactors in the middle. So the question is, if we leave out the enzyme itself, can those cofactors still do some of the work? Can we connect on? Also, can we connect these cofactors to geochemical reactions as well? So the question is, can we use cofactors, especially organic cofactors, as a missing link between geochemistry and biochemistry? And could they be part of pre-enzymatic catalytic networks as well? That is also based mainly on amazing work by Joanna, as you can see on the bottom left. So just to explain a little bit more the background, the reasoning behind it, it's basically imagine a rock pour or an environment of your calling. Basically, you would have first simple organic fixation, CO2 fixation, or at least you would get some type of organics. You have mineral surfaces or also ions in solution that could do those that could catalyze these reactions. Then in the next step, let's just assume for now, because that's not from Ferté, I'm not a synthetic organic chemist, but we could come up with more complex organic molecules like that. The question is, why would some of them become organic cofactors with a certain task and others wouldn't do the same thing? So there must be kind of a selection process that might have been constrained by the actual environment. So under which environment does a redox cofactor, for example, take a hydride and give it away, and does give it away again to another molecule, which is depicted here. What we started experimenting with was NAD, to see could we actually just reduce NAD under geochemical conditions. Also one step more, there's just an amazing preprint out by Joseph Morin's lab, where they're basically showing that this is possible as well. So if you have NAD agent solution, you can actually even stereochemically actively reduce keto acids. They do it in solution, but it's a very same principle that is being shown here. So we wanted to say, can we reduce NAD plus to NADH? And this was the FINA's first paper. We did mineral-assisted NAD plus reduction. I'm sorry that I have to rush through it like now because it was really, really interesting work, but it's published, so you can look it up. So basically what the FINA did was dissolve NAD in solution and different buffers with different metals, and looked if she could actually quantify, see NADH in the end, which she could. And she also found the biologically active version of NADH, which is 1.4 NADH. And the good thing is you can look at it via protein NMR quite easily, and also you can quantify it quite easily. So what she found out back then is that both iron, nickel, and also cobalt, but I'm not showing that here, can promote the reduction of NAD+, but they do it in different ways. As you can see, we did different reactions under hydrogen or argon, and for iron, it's not that important if it's there or not, the hydrogen in the atmosphere, but it works faster. And nickel, for example, is a true catalyst for the reaction, not only something that also gives electrons, such as iron. You really need hydrogen for nickel to work, for example, for the same reaction. So again, please look that paper up. It's really nice to read. I think it's also quite accessible to read. Since then, we tried to approach more natural systems. So Defina, together with Chuche from BSI from Mülheim, was working on different mixtures of nickel and iron to figure out what does every metal do, basically. We also got more natural systems when it comes to buffer. We decreased the buffer concentration. We also decreased the ratio of metal to NAD, and it still is working, which is very nice to see. And we also see quite some differences depending on what mineral or metal we are using for this reaction, and which right now trying to figure out what that actually means for the reaction itself. So to sum up, I want to say or convey that it's possible to connect organic cofactors to mineral surfaces. The question is, does it tell us something about the selection of certain biomolecules? And the question is also for us now, is it possible to create a non-enzymatic reaction network based on these type of cofactors? Last but not least, I just want to tell you, what are we doing now? What are we planning? So we are really trying in our group to connect a lot of different disciplines under one hat, so to speak. So microbiology, biochemistry, bioinformatics, geology, geochemistry, and of course, full-on chemistry, just with cofactors and other organic molecules as well, from the bottom up and the top down. But this science wouldn't be possible without the people. So this highly depends on my students that just started, and also on all the amazing collaboration partners I have in Marburg and also outside of Marburg. And with that, I apologize for rushing and taking so long, but thanks for listening to me, and thanks for inviting me as well.
Well, I can clap.
Silence,
Thank you, Martina.
yes.
Yes. Thank you for your insightful talk. And yes, so what we can do is we can take questions for both Martina and Alex at the end. It's much more
Yes.
convenient. And since we are pressing on time, we can move to Alex. Alex, are you here?
I am.
Yep. So
Can
this
you
will
hear
be
me?
a very nice, this will be a very nice transition from Martina's biogeochemistry to Alex's chemical autocatalysis, which Martina was trying to talk on. And this is why we chose these very different topics, which are different yet very interconnecting in OoLEN. Alex, can you share your screen?
Let's see. Can you see it?
Yes, I can see it. So
Okay.
before moving on, I'll just introduce you a bit. I'm sure you'll introduce yourself, but being a co-host again, I need to make some comments. So Alex Blokhuis is currently a postdoctoral fellow in the Wessing and Sijbrinotos group in Netherlands, and he'll be moving to iMedia Nano, if I'm correct, in Spain, Madrid real soon. So, yep. Go ahead.
Well, hello everyone. It's an honor to be invited to the OoLEN seminar. Very admirative initiative, like it a lot. Today, I want to talk about some interesting phenomena that emerge from physical chemical processes. I think these are interesting because they help us to develop some mechanistic conceptions of how life can emerge, a conception that we're all still seeking for in OoLEN, and I hope that this contributes. This is the paper that it's about. So in the end, in my view, our scientific task in Origins is to explain abiogenesis, the hypothesis that non-living matter can give rise to living matter. And now clearly, there is a lot of complex behavior that abiogenesis must ultimately explain. Let's just have a little look at some of the things that living matter does. I mean, we have cell division, metabolism, homeostasis, genetic replication, photosynthesis, controlled transport, I need to test. You can make a very long list of these things, and I'm not even a biologist, so I'll stop here. But clearly, we have a daunting task before us, because in prebiotic chemistry, we don't have a lot to work with. We have simple physical chemistry. And if we want to assemble these increasingly complex objects and tasks and properties, these are the basic ingredients that our ostensible soup of chemicals can do. Like we can do some adsorption, diffusion, evaporation, precipitation, elementary reactions, very simple stuff that matter intrinsically to us. But clearly, for non-life to explain life, these processes somehow must make more complex ones possible. These more complex processes should somehow emerge. But let me first start by pointing out that this sort of emergence is, in a way, already natural. It's already something we implicitly do in chemistry, for instance, in autocatalysis. So all autocatalysis in all of chemistry always can ultimately be decomposed in uncatalyzed reaction steps. That's a function of the definition. The IUPAC definition makes it that this is true. And so in this sense, all autocatalysis always is emergent with respect to small reaction steps. All autocatalysis is network catalysis. And so starting from elementary reactions, we suddenly get this capacity to make more of something. So clearly there are already interesting properties that you get from physical chemistry, but we want to push that a little further. So now for abiogenesis, one idea is to imagine that just this sort of process of emergent properties popping up many times over, and each time enabling new emergent properties, making it simpler to do more interesting stuff. That's one conception to address the problem. And today I want to talk about some properties that autocatalysis will unlock. And for that, I want to start with a thought experiment. So our story starts in oil. In this oil, we have a droplet of water, water compartments. And in this compartment, we have some small molecules, formaldehyde to be exact, but I will refer to it as C1 shorter. Now, this compartment is particularly lucky because there's something else in there, glycoaldehyde, which I will call C2. And this allows it to do this. C1 can react with it to form C3, which can in turn form C4 and so forth. And starting at C4, we can break it up again, according to this scheme, to make more glycoaldehyde or more C2, giving ever more molecules to participate in the cycle. So after some time, the droplet will look like this. Now imagine that we had a neighboring droplet that was not as lucky, that has no C2. And now we're getting something interesting, because surely now we have concentration gradients and osmotic pressure differences. So our small molecules and our water can now traverse. And clearly the chemical potential gradient is such that in a while we will have a current of both small molecules, and we get this. So now a small guy gets stripped of all its valuable resources, and we see that we started with autocatalysis and with some physical chemistry like partition reactions between water and oil, some diffusion, we suddenly get growth and some rudimentary form of competition, if you allow that term, but I'm not married to the terminology. So we can conceive of simple model systems that do these things with some characteristic timescales, and we can make phase diagrams for that, and then we can even see where would volume doubling occur. Now, a simple critique here is that foremost reaction does not really look like that. If you are a chemist and you look at that toy model that I showed you, you may recognize that we are using the foremost reaction, and chemists know that the foremost reaction clearly involves a lot more species, a lot more reactions, and a lot more side reactions. Is this going to pose a problem for us if we actually want to talk about chemistry in the real world? Well, chemists also know that much of this complexity is very inconsequential. For a physicist, that means that we can come quite far with very low dimensional, very simple, effective descriptions. We can understand a lot of this system. So one thing you see is you'll have few rate limiting reactions, because not every reaction can be the slowest one. That means we have a lot of local equilibria. We have many species that are barely there and reactions that are not occurring all that much. A lot of species have qualitatively similar effects on osmotic pressure. And actually a lot of species cannot traverse the droplets very well. So in the end, you can actually understand a lot about the system with lower dimensional descriptions, even though you don't capture everything. Of course, the more we know, the better we can do that, and the better we can justify the simplifications. But here is the biggest issue of all. To this day, foremost is not understood. No one has ever come up with an acceptable quantitative description of the foremost reaction, and it's not likely that it'll happen soon. For our work, that is not that big of an issue, but the day foremost is actually understood, we should definitely go back to this. And this is something that me and quite a few colleagues are working on to actually understand foremost. That's something for another day, but that will change this picture. With all that being said, it will turn out that this simple description that I provide here actually does tell us a lot if we attempt to do something like this in experimental practice. Here's some detail about that model, but it's in the paper, and this will make more sense if I've presented the rest, so I won't dwell on it right now, but here you see it's very low dimensional. So let's move to experiments. We can make a perfluorinated oil phase with neatly neighboring droplets to mimic our thought experiment. So here you can see at the 0 hour and 4 hour marks what that can look like. So we actually have access thanks to this prior work from 2016, to a neatly 1D system that's quasi-periodic to do the first part of the thought experiment. Now, I have some videos to show how that works. So we have something that gives us monodisperse droplets, and then we also can push them in chambers and channels so that they can be neighboring, and so that we can actually have different populations that we alternate. And this allows us, in turn, by video microscopy, to monitor the volume of neighboring compartments with alternating compositions. So we just make a lot of these videos that we follow over time. And here you can see the volume measurements for different types of compositions. So this is the type of setup we use. Apart from performing our thought experiment, this also provides us with all sorts of other experiments we can access. For example, all sorts of control experiments and measurements of permeability. That's important because then we can check if our description of the transport properties make sense, and what components transport, etc. So for instance, it allows us to look at the functional dependence of transport coefficients, which to leading order scale reciprocally with the distance. But the problem is quite a bit richer than that, and clearly this approximation works a lot less well for very close distances. But so we can characterize these things. We also looked for low dimensional descriptions of the chemistry, as I mentioned, and to monitor some actual chemistry to compare it with, we notably did Nash reactions every minute, or even every half minute, I think, and then compared that with UV-Vis. We also did NMR experiments. We also did serial transfers, just to have quite some data to compare it to in different regimes, and also to characterize the different site reactions. So for example, one important one that's well known is the Cannizzaro reaction, which turns out to not be as important as I thought. You can actually leave it out of the minimal model, but you can also include it, and it doesn't make a very big difference. So if we put all of that together, we can effectively recover much of the 1D behavior. As I said before, there are considerable simplifications. There's a lot we're not capturing, but we can capture the qualitative behavior and the volume effects. And this is what we were looking for in this study. Okay, with that being done, we can now move to something a bit more complex, which is a two-dimensional instantiation of the system. In this video, you can again see two droplet populations, but since it's 2D, we now need to make it a bit more clear. So here we have marked droplets that have no catalyst initially with green fluorescent beads. So these are essentially food droplets. And for these droplets to grow, like the colorless one, they need to have access to the food from surrounding droplets. And so what you're now going to get is that your surrounding is going to be very important. And this will translate to differential growth, as you can also see in the histograms on the right, where there's clearly a large distribution of sizes that pops up. The variability in growth strongly correlates with the neighborhood, that is the number of surrounding feeder droplets. And more particularly, we have a monotonically increasing function overall. And if we put more food in the function, then also this increase is stronger, which makes sense, but so we can quantify that it indeed does that. We can also mix several catalytic droplets with different amounts of initial catalyst, and we can test them in isolation and combined. And now you can also see that there's interesting competition effects, like the one that's more enriched in catalyst can actually grow a bit better in this case, and the one that has less of it actually does a lot more poorly than in the isolated cases. So there are some effects that one could describe as competition. Then there's also, of course, a lot of spatial heterogeneity initially, but also during the competition, which makes the analysis a bit harder here. So over time, the position may change and the surrounding may change. So overall, the problem becomes quite rich, but I hope I have illustrated the key points here. So we have thus far seen growth, but after the growth, you'd like to do something with that to get something like a notion of fitness. And we can do that here using shear forces. So we can use a single channel, and that gives us a single division, but we can also have another successive division channel, and in that way, droplets that grow to 4 times their size, which was not possible in the 1D setup, but which is possible in the 2D setup, we can multiply them. And in that way, you actually get an emergent form of fitness, if you allow that term. So that allows us to also grow droplets, split them, and then put them back in for another round. So we can basically regrow them. And we find that it is indeed possible to actually do that. So here are some more videos where we do exactly that. And here, one of the interesting effects that you're now getting is that there is a positive feedback between the growth and the size. So the droplets that are already big more easily get bigger, and this basically becomes visible in this sort of experiment. And basically this type of positive feedback between growth and size, that gives you a very weak notion of heritability in the growth. So clearly it's not heritability in any sense that we would like, but there is already this. So we have some weak notions of heritability that emerge just from this sort of process. And theoretically, if we study this process of growth and then splitting, we can numerically find stable protocell cycles. So in principle, we could push this further and really sustain these things. We also had an analog version of that where we just tried bulk serial transfer of the foremost reaction. I think this is not in the final version of the manuscript, but it might be on the preprint. But basically, yeah, if you just leave the foremost reaction in a single vial and you wait, then you get this brown stuff. But that won't happen if you keep transferring to a fresh solution, and this eventually reaches a steady state. So with that, I'd like to conclude that we find that starting from autocatalysis and coupling it to compartments with simple physical chemical processes, we managed to get a lot of other interesting emergent properties. So we get growth, we get reproduction, we get notions of selection, and we get forms of variation. But clearly, it's not there in a sense that we can do Darwinian evolution. And in our view, one thing we clearly lack here is a diversity of heritable states. So clearly, we're not there yet at all. But what it does allow us to do, at least, is extend our conception of where interesting replicating compartment properties can come from and how much we actually need in terms of complexity or miracles from the outset, and how much just pops up eventually if you have the physical chemistry to start with. So now I'd like to move to the second part of the talk about career advice. So just half a line about me. I started as an organic chemist, then I started to do more theory, then I moved to theoretical physics, and then I moved back and started doing origins, where you can really make use of everything. Now a disclaimer, I'm still a postdoc. I'm still figuring out the career thing myself. So how much advice can I give you? Well, one advice that I can give you is I can give you advice on how to figure things out, because that's where a lot of time will go when you try to figure out the career thing. So at least I can help you figure out how to figure things out. And that's probably something that saves you a lot of time. So when I tried to figure things out, I started attending a lot of career advice talks, but actually, yeah, there's some bias in the type of careers that get selected for with these advice talks, and they were typically not discussing the career as I envisioned it. I knew a lot of people that did not have a career like it was displayed in those talks. And basically, there were quite a few scientists that I admired with a career as I envisioned it. And I just reached out to those. Not actually all of them. One of them I really wanted to talk to, but I never did. But he then became my boss. So in the end, it all worked out. But this is the one thing that clearly worked for me. Reach out to people that actually do what you want to do. Not all scientists do what you want to do. What do I mean with that? Well, here are some very relevant questions if you're thinking about how do I see my career in science, if at all. So for example, what kind of research do I want to do? With how many people do I want to do that? And what do I need for that? And what do I want my role to be in all that? And of course, that also means that you decide what you don't want to do or what you don't like your role to be. Like I love doing organic chemistry, but today I prefer just doing lots of calculations and doing a few experiments. Clearly, this is not what I want to do full time. Deciding on what your answer is to these questions is very crucial because on very different places on the planet, the very different careers are possible. So when I'm talking about research culture, like, are you not actually seeing your boss all that much, or you're spending 2 days a week doing calculations and exchanging math books? That will be very different depending on the country, mostly. What is the funding structure of your science? Can you actually maintain a group? How stable is that? What type of positions do you have? Can you be a technician, or can you be quite shy and still do interesting work without leading a large group? And of course, there's a lot of expectations and conditions tied to these positions, and these are very dependent on the country. So for instance, the researchers that have the type of positions that I'm interested in, they mostly live in France and some other European countries. In the Netherlands, for the reasons I just mentioned, this is barely existent at all. It's just a lot harder to do the type of career that I envision. And so the career talks that I followed in the Netherlands were not very informative for me. So very succinctly then, my advice is this: there is no silver bullets because everyone's different and we're looking for different things. You need to figure out how to figure things out. If you figure out what you want and where that's possible, and vice versa, then you already made enormous steps, because for a lot of these different places, you need to invest quite a bit in your network, in your time there, in the people you know, before you can actually do something there. So make sure you figure this out as early as possible. Don't be like me and wait too much. And make sure that you reach out to those who can help you with that. And with that, I think that's all the advice I can give at this stage. Thank you very much for your time.
Thank you, Alex. That was something. Okay, so I think the floor is open for questions for both Martina and Alex. We can do that in the chat. You can raise your hand. You can open your mic and embed video. You can ask them. Whatever.
Should I stop sharing?
Yes, you can. I think we have a question from Sean.
Hey everyone.
Hey.
Really nice talks from both speakers. Well done. They were really great, and great advice as well. I just had a question for Martina. Just about the work you were doing on Rainbow, and looking at modern hydrothermal systems. Could you talk maybe a little bit more about how or if you're linking that to kind of Fidean systems, like based on the difference in chemistry and stuff?
Yes, I will try. Thanks for the question. Hi, great to see you again. Although it's far away, but still.
Likewise.
So I think the shortest possible answer is, if I understood correctly, because you were a little bit robotic, but that might also be my connection. is that, I mean, we both come from like an alkaline hydrothermal vent background. And for me, it was kind of mind-blowing, although it was not a secret, right, that there were serpentinizing systems that were not alkaline. And as I'm really interested in what hydrogen has to do with proto-metabolic reactions, I just try to become more agnostic on that front. I also think that certain chemistries are like, we started some work on riboflavins, for example, as cofactors, and they usually work better, for example, in more acidic conditions. So basically what I'm doing is just opening the palette a bit to basically have more options as well, not try to like focus too much on one side, especially when I was in Utrecht. That is just, I don't know, I don't have any paperwork on that really, but actually a lot of geologists told me that it's very hard on white smokers, you know, because that to have such cool systems might have been hard on very early Earth. So what I'm envisioning, also I have forever I wanted to write a review or perspective on that, but maybe there's even a opportunity to start from acidic hot ones, because chemically they're kind of connected, and the further you go away from a hot spot, you would get the opportunity to become alkaline and cooler. So maybe there's even a connection between the two systems over time. I hope that answers the questions a bit, but yeah, for me, what I did at Rainbow was really disconnected from origin of life. But it was also interesting to see, because the chemistries that come out there when it comes to just CO2 fixation are quite similar, actually. Although, of course, a lot of iron dissolved, etc. But what you can do is on a simple level, like simple synthetic level, quite similar to what you can do in alkaline vents. But when it comes to more complex networks, etc., I think we cannot say that we can work at 350 degrees anymore.
Yeah, that's really interesting. And just on the advice on early Earth and super hot systems, I think you can probably put a geologist on a scale of temperature between where they think it's really hot and really cool. So yeah, I think we're still figuring it out. It's nice that you're looking into all aspects.
Yeah, I'm just trying not to get too much in my head about it. Thanks.
Martina, thanks, Sean, for asking that. I think we have Rio. You can.
Yeah, thank you both for the great talk. And I have a question to Alex. Yeah, good to see you again, Alex. And so your system is very neat, I think, and you successfully combined autocatalysis with the growth of the compartment. And do you have any ideas of combining the autocatalysis with the division of the compartment? Because I think that's necessary for Darwin evolution, right?
Yeah, nice to see you again, Rio. Thanks for your question. Combining it with division. So I think it has been done to some extent in Nijmegen, or at least there are these coacervate-like systems where fibers grow, and basically there's some instability that then makes it split in two. So that comes quite close to it. We did ponder a lot about this, like we've had this discussion for years. Okay, shear, that's quite a basic ingredient, but still, can we make it more autonomous? We didn't find a better method to do it. We did have discussions on somehow other conceptions, because still here we are very much tied to this idea of a protocell, right? That somehow you need some sort of protocell that needs to grow and then divide. And maybe that's too much of a restriction. Maybe those things never existed. Well, that's a bit of heresy, but I'll say it anyway. But for example, if you look at certain sorts of reproduction where maybe you have a seed and then it grows somewhere else and then it pollinates again, like you can chemically absolutely conceive of things like that. So definitely we can think of autocatalytic processes that just don't start out with a compartment splitting in two, but that spit something out and then make the compartment anew somewhere else. So, well, there is some stuff about that at the end of my thesis, but that's all I can say about it. That's we can extend this idea. There's a lot of other mechanisms out there. But for us, this was quite a pain to come up with anything to split the droplets other than with shear forces.
Okay, yeah, thank you very much. Thank you very much.
Yes, Yannick, you have a question?
Yes, yeah, thanks. Thanks, Alex and Martina, for both the talks. It was super cool. Alex will get in touch anyway, so I'll ask you something later then. Martina, was super nice to see your Vita, which is kind of unusual. So I didn't think you were a lot into science communication before. It brings me to a question I honestly wanted to ask you already last week, but then I didn't come to that. That's how was it for you now to become a PI? Especially after the short postdoc experience. You were just 1 year in the Netherlands, right?
Yes. So not a little bit more, but yeah, by the time that I realized or I got the appointment, it was like barely a year actually. So that's a great question. I also have tried to come around that. And actually that's also a question that I was asked in the selection symposium a lot, like from different people. So like, why should we take you? You have like so little experience as a postdoc. My counter argument was that it really depends also, I think, how your PhD goes. So if you have a PhD that is where you have to be very independent already, which I had to be, and I had to build up a lot and organize a lot by myself, I think that can make up to a certain extent. That doesn't mean, honestly, I would have been happy another 2 years in Utrecht also because I kind of liked the Netherlands and also the work I was doing there. And also actually being a bit away from origin of life, a bit was also nicer to take. You can always, I think, benefit from that. But I also think having a little bit of experience outside of academia helps in, I mean, not, can I say leadership? But yeah, at least you have a different view on some things, I think. So there are several ways of getting experience. Let's put it like that, I think. But I also have to say, it's probably not the most common route. I was also surprised that in the end I got the position, if that helps in any way. But I think at least I made the point that experience outside of academia also kind of counts. And that's also something that you can provide to people when they're part of your group.
How was it to switch from the role of postdoc working in a lab to supervisor?
That's also a good question. I'm still figuring that out. I have to admit, that's the one thing. Being a postdoc is actually quite nice. If you have like a good contract, you know, that's like more than half a year or a year. I found it at least I found it very enjoyable. But I'm also someone, probably because of my background, I tend to be not that worried about non-permanent positions, just because I thought, well, all is going to be fine. Worst case is I do something different. It's all good. It's all good. I like the transition, but it was a little bit harsh because I had to do it quite quickly, you know. And I think maybe some of my students are watching it on YouTube. They might be able to tell you how I'm doing with the switch, but there are some times where it's hard. But concretely, how I try to organize it, you know, is really to plan the projects as much as possible, but also include people in the in making their projects as well. So they also already find independence quite early because it helped me quite a lot. So that's how I'm trying to do the switch a bit, you know, like they help me leading as well, kind of in that way. I'm not sure if there was a concrete enough answer, but as you can tell, I'm still figuring it out, but I'm trying my best to include everyone in the actual process as well.
Yeah, I guess so. You started also just less than a year ago. So it was also, I had the same question to Claudia Bonfio, who is doing this for already 3 years. So that was then already see a bit of a difference, but it was insightful. Thank you.
Yeah.
Thanks, Yannick. So we have a question for Alex in the YouTube chat, and thanks, Lauren, for managing the YouTube chat for me. I can't keep my eyes at two things. But hi, I think Martina from Centro for Astrobiology in Spain, in Madrid, Spain. She asks that I'm a PhD student from Centro for Astrobiology in Spain, and she has a question regarding water droplets in oil. You explained that autocatalytic encapsulated cycles can compete, but why food precursors would enter the compartment with a higher rate for autocatalytic than for non-autocatalytic? In summary, could you explain in more detail the mechanism behind the osmotic flux of precursors and metabolites?
Sure. Yeah. I think I understand the question. So yeah, why does the food go to one droplet and not the other? So the idea here is that this reaction is quite favorable. So if you have this formaldehyde or its hydrates and you can do this autocatalytic cycle, then actually most of it will be used up to make other species. And in particular, most of the higher sugars, they're not so mobile in the perfluorinated oil. So once you start making those, it's basically stuck there. And it's then only the really small species that can effectively be exchanged. And now if you compare it with a droplet that just has all this stuff still lying around unreacted, then the net transports will go from the droplet with a lot of this stuff to the one with not a lot of this stuff. But also, since you're putting a lot of molecules in fewer molecules, you're also getting that the water wants to go there. So this is really where the autocatalysis becomes important, that you can do both of those. So you could also actually compare different types of chemical situations and how much growth you can get from them. In my thesis, I have a lot of those compared, and autocatalysis is really special in this regard, that you can actually completely consume the other droplets. But it is because of this reason that you basically store everything in these larger sugars that are immobile, and then you use up the other stuff from the neighbor. I hope this clarifies the question.
I think we don't have any more questions, so I can ask a final question to Alex. In your system, how were you tracking the foremost progression in your droplets? Were you tracking it with dye, or does the yellowing point fluoresce under specific conditions? How were you tracking that?
So in the droplet system, we could only really monitor it with the volume. We had little other to look at. In some earlier versions, we had some other signatures because in the end, we opted for a base that can also traverse TMG. Initially, we had a base that actually crystallizes out, so there were a bit more things you could observe. But yeah, this is why we also did bulk experiments in NMR or in tubes and with UV-Vis. Those don't perfectly translate, but we're looking for a description of the bulk droplets for the chemistry, and we're looking at those experiments. So yeah, that's the comparison we make, but definitely this is one thing that we were always hoping to get at some point, some more access to what's really happening in the droplets and if there is any difference.
All right, I don't see any more questions in the chat and not even in the Zoom. Thank you, Martina. Thank you, Alex, for joining. This was really insightful.
Thank you very much.
Yeah, thank you
Yeah.
for the opportunity. It was a pleasure. And also everyone who got up very early. Thank you.
Oh
Especially
yeah,
Alex, I
yes.
guess, at this point. I hope it was worth it.
Yeah, absolutely. Totally.
Okay.
Thank you for the talks.
Bye, everyone.
Bye-bye.
Bye-bye. See you next time.
See you next time. Thank you.
So you stopped the YouTube live?
I'm gonna end the stream now. I think yeah. Okay. Hey, see ya.
So you stopped now. Okay. Do I have to do anything?